Welcome to ISL Enabled Virtual Lab of CSIR.
Hi everyone,
I'm Utsuka.
I am Jigyasa and
you're
you are listening to India Asks Why,
a science podcast supported by India Bioscience.
where we do the research for you to get smarter.
Join us as India becomes more curious,
One question at a time.
One evening, when Utsuka and Jigyasa were enjoying the winds at their window, it suddenly started to rain. Along with it came the sweet smell of rain, which made them wonder, why does rain sometimes smell so good? While exploring the reasons why, they find out that a chemical produced by a specific bacteria is behind the smell. In the second segment, they speak to Dr. Samay Pandey, a researcher who studies bacteria, to know more about fascinating chemical molecules that the bacteria produce.
It's that time of the year again. Monsoon and lots of rain. Doesn't it feel so good to hear the raindrops hitting our roof and windows?
The good smell of the rain brings me back so many memories. What is the smell called again? Petrichor, yes.
Petrichor? That sounds poetic. I wonder why rain sometimes smells so good.
So, the smell of wet earth is not because of rainwater itself, but because of various chemical molecules. The main one is called geosmin, which is produced by spores of a bacteria living in the soil called streptomyces.
Wait. That's a lot to unpack there. So many big words. Geosmin, streptomyces, spores, and bacteria. Go slow and tell me everything.
Okay, I'll slow down. So, streptomyces is a spore producing bacteria that lives in the soil.
What are bacterial spores? Are they different from bacterial cells?
So, Jigyasa, spores are tough structures that are dormant, meaning they are not performing any lifelike activities or consuming nutrients. Spores help in dispersion. They can move from one place to another to.
But why do these streptomyces bacteria have to produce spores to move from one place to another? Can't the normal bacterial cells move?
No, streptomyces is very unique. Unlike other bacteria, which are single celled or have special structures which can help them move, streptomyces cells are joined together to each other to form a thread like structure. These threads are entangled and run inside the soil.
But first tell me, why do these bacteria have to move places?
Well, imagine yourself as streptomyces, okay? You are stuck at a place that is soon running out of nutrients. What would you do?
Yeah, if the conditions at the current residence aren't favorable, I should be looking for a better place and move out, shouldn't I?
Exactly, these bacteria love food. So that's why spores come into picture. Streptomyces threads give out a special structure called hyphae that is made up of multiple cells. Hyphae grow out of the soil's surface, which gives rise to multiple spores that can then get dispersed. So you can escape out of that unfavorable condition.
So, bacteria make these spores on their hyphae for dispersion. Okay, that makes sense. But what has this got to do with that geosmin?
Geosmin. Yes, so these bacterial spores are sitting on the hyphae and are looking to hitch a ride to a different place, right? So they are stuck in the soil and can't move.
They need a vehicle of some sort, right?
Exactly. So that's why the latest research says they produce geosmin to attract their vehicle. The vehicle here is called a springtail, which is a teeny weeny six-legged animal.
Whoa, I'll look its image up online.
So I was saying that geosmin that Streptomyces spores produce attracts the springtail. The springtail thinks it is food and comes to feast on those bacterial spores.
Wait, you said springtails are like vehicles for the bacteria to hitch a ride. Now you say that the spores get eaten?
Haha, don't worry, don't worry. So bacteria produces spores in large numbers. So while springtails are eating these spores, some latch onto the surface of springtail's body. And when the springtails move to a different place, they drop off the spores that was once stuck. Some spores even make it out of the springtail's digestive system unharmed and get dispersed that way too. And then the spores restart their new life at a new place. That's pretty cool, right?
That's a very interesting interaction between springtails and streptomyces. But what I still don't understand is if geosmin is produced in the soil, how does it reach my nose?
So, when the rain hits the soil, at the point of the contact, the larger water droplets form smaller air bubbles which rise upwards and burst into tinier aerosols. So, do you know what aerosols are?
Yes, aerosols are the solid or liquid droplets suspended in the air, like the droplets that come out of the deo bottle when I spray it on my body, right?
Yes, and these aerosols contain geosmin and other aromatic elements which give petrichor the smell. The wind then carries this aerosol to your nose.
This makes me wonder, in a different condition, would the bacteria produce a different molecule and would it do something else?
Well, you've got to hold on to know the answer to that question, Jigyasa. We are going to ask Dr. Pandey in the next segment about this very same question. So stay tuned.
And now it's time to ask a scientist. Hi everyone, getting back to our second segment of the interview, we have with us today Dr. Samay Pandey from ISE Bangalore. Utsuka, why don't you go ahead and ask your question to Dr. Samay Pandey?
Okay, so Dr. Pandey, we just learned that geosmin, which is the chemical behind the smell of the rain, is produced by a bacteria, and that it is produced to attract springtails to help for its dispersal. We also learned that geosmin is what is called a secondary metabolite. So can you talk to us a little bit about what secondary metabolites are and why do bacteria produce them?
Well, that's a deep question, but now there are compounds that many organisms make that are not necessarily giving you any growth benefit or energy, right? So for any life form to function, you need energy. So that's why you make many compounds. Some compounds will give you energy with the primary metabolites. Some compounds you produce them, but there's no obvious reason why you would produce them as a bacterial cell because it's not giving you any energy. So those are secondary metabolites. Okay, now what are different types of secondary metabolites? Right, so I'll just give some examples. And I think probably one of the ones that I would highlight most importantly, which has probably changed the course of human evolution, to put it mildly, I would say, is antibiotics, right? Now, another example of secondary metabolites can be what is now famously called as quorum sensing molecules, right? So these are the molecules which are used by bacteria to talk with each other, to communicate.
So how do you know that these bacteria are communicating with each other with this secondary metabolite and so on?
We in our lab are interested in sort of more of why questions. So we are interested in then why should you be social? Why should you communicate with other bacteria? Let's say, why should you help each other? So we are interested in why questions. So what we do is something called experimental evolution or evolution in a test tube. Now,
Wow.
think of an approach which will allow you to go back and forth in an evolutionary timeline, right? So the simplest way one can do is, and at least the simplest experiment that we do in our lab is, let's say we're working with a bacteria that makes a secondary metabolite, which kills other bacteria, right? And we know that making this secondary metabolite is very costly. So what we can do is we can grow this bacteria that makes secondary metabolite, have another bacteria that is now susceptible, that means can be harmed by this secondary metabolite, put them together in a test tube. Okay. Now what these bacteria do is they grow and they make multiple copies of themselves. Then we transfer fraction of these populations. So let's say out of 1 billion, we transfer 1 million to new test tube with fresh food, right? And we keep doing it every day in our lab. Okay. Now we transfer 1 million to new tube, but remaining almost billion cells, we also freeze them in -80. Right? So -80 means that's a temperature that we are talking about, in which bacteria can stay alive without changing their biology. Right? So after, let's say, 50 days of doing this experiment, we have something which we called an evolved bacterium, which has changed itself to acquire new ways of behaving with other bacteria, either harmful or helping, and all the previous ancestors as your frozen fossils. So you understand the change of biology. You can go back and forth in this evolutionary timeline to understand how an aggressive bacterium, which makes a secondary metabolite, is changing itself to make that metabolite more efficient in killing whatever it wants to kill or harm. And at the same time, the bacterium that is being harmed, how it is changing over time to protect itself better. And we can change that. So that's how we study these behaviors.
Wow, so you're basically studying a chemical warfare inside a test
Correct.
tube.
Perfect. So we're studying evolutionary warfare or chemical warfare, as you correctly pointed out, in a test tube. Though we are interested in questions of why, but that why has a mechanism underneath it, right? So why will a bacterium do this or a cell do this? It's a great question to ask, because it will understand us basically, to figure out how the life works on our planet.
That's super interesting.
This is so fascinating. Yeah. So why does the social interactions that among bacteria or the evolution of bacteria interest you so much?
So what interests me most is the evolutionary process and social evolution in general. And this started off with a very simple sort of a question that I couldn't solve, and later on realized it's probably one of the cornerstones of evolutionary biology in general, is why should anything help anybody else? Right? If an organism is helping another organism, and I mean by helping, I mean actual helping, you're investing your energy or resources, right? In theory, those energy or resources can be diverted towards making more offsprings, making more babies of yourself. So why wouldn't you make more babies of yourself, which is sort of the fitness, as Darwin said, evolution of the fittest. So why should you invest your energy, resources in helping somebody else and not help yourself to basically be successful evolutionary? Right? So this was a basic question that fascinated me. And then I realized microbes are an easy and more convenient model organism to ask these type of questions.
Coming to the end of it now, we would like to ask, what is this advice that you would like to give to our listeners on curiosity as well as taking science as a career, maybe in the future?
I think at the crux of it all is, if you're curious, just try to answer your curiosity and you'll automatically develop into a scientist. So you don't have to be in the lab. You don't have to be associated with active academia. As long as you started asking questions and trying to find its answers based on available information and not, hypothesis, just basically predicting something, but rather relying on information that you have in your hand, you're already a scientist. Right. That's it.
Yeah. Jigyasa and I always love asking questions, and it's good to know that we are already becoming budding scientists, right?
So, Jigyasa, Do you now understand why rain smells so good sometimes?
Yes, the rain smell is due to a molecule called geosmin, which is produced by spores of a specific bacteria living in the soil. They make this molecule to attract their transport vehicles, the springtails. When the rain hits the soil, they produce tiny aerosols containing geosmin that reaches your nose when the wind blows.
Yes, and Dr. Pandey spoke to us about many other molecules like geosmin, which bacteria produce. He also described super interesting evolution in a test tube experiment he performs to study why bacteria produce many such fascinating molecules.
True. But listeners, what we know about geosmin and rain smells may change over time as we get more evidence.
Who knows? One of our listeners might be interested in finding out more about the role of such cool molecules like geosmin. So, that's it for today. We would like to thank Dr. Samay Pandey for being on our episode.
If you want to know more about today's scientist, check out their profile. The link is in the show notes.
If you have any questions that you want us to explore, shoot them away to indiaaskswhy@gmail.com.
Or if you would like to directly talk to us, join the fun on our Telegram group. Again, the link for it is in the show notes.
And for more updates on the podcast, follow at India Asks Why and at India Bioscience on Instagram and Twitter.
Shweta N. Hegde and Ruchi Manglunia are the hosts for the podcast.
Ira Zibu edited this episode of the podcast and transcribed the interview segment. India Asks Why is supported by India Bioscience's second outreach grant and is hosted on India Biospeaks.
So stay tuned and stay curious.